Hello everybody and welcome to Molecular Genetics and Gene Regulation. Today we're going to be talking about DNA damage and repair. This presentation is based on an atlas page by Isabel Conrad, Catherine Floyd, Kelsey Hearing, and Mackenzie Watkins. So you might be wondering, what is DNA and what damages it? So first let's go over DNA. DNA is a double helix made of two strands that are made up of a phosphate and deoxyribose sugar backbone. These are connected by nucleic acid bases. These bases contain adenine, which double bonds to thymine, and cytosine, which triple bonds to guanine. Now let's go over damage. One of the biggest causes of DNA damage is the formation of pyrimidine dimers. This occurs from UV light from the sun. When the UV light hits the DNA strand, it causes the pyrimidine bases in the DNA to bond to each other. This formation prevents essential machinery from binding to the DNA. One of these proteins is the DNA polymerase, which is essential for replication. Pyrimidine dimers are just one form of DNA damage. One of the most dangerous forms of DNA damage is the presence of free radicals. These free radicals are of the oxidative species and are extremely reactive. These free radicals are not completely filled by their electrons, so they try to take other atoms' electrons to fill their electron cloud. This leads to extremely unstable DNA and eventual damage. Another form of DNA damage is actually very common. If you've ever had an X-ray, you've potentially been exposed to DNA damage. In an X-ray, ionizing radiation is sent through your body to develop a picture. This ionizing radiation can actually cause single or double-stranded breaks, and if left unrepaired can lead to very detrimental effects. So how do bacteria fix this problem? Bacteria actually call for help. They use what is known as the SOS response. In this mechanism, they use what is known as an operon. This is a long strand that contains multiple different genes. On this operon, there is a promoter. Downstream of this promoter are actually genes that can help with DNA repair. So first let's look at what happens when there's no DNA damage. LEX-A will bind to the promoter, and another protein called REC-A will be inactive. Because LEX-A is still bound to the promoter, there will be no translation of these helpful genes. This means that this operon is off. Now let's look at when there is DNA damage. REC-A will now be active, and this protein actually cleaves LEX-A, which doesn't allow it to bind to the promoter. This allows for the operon to be transcribed and translated, which means that it's on. As we said earlier, the SOS genes are translated into proteins that actually help with DNA repair, which is what we will discuss later. If you thought that was complicated, what do you think trees do? Or people? Or even your pet fish? Now we'll discuss how eukaryotes deal with DNA damage. If you've been paying attention, you might wonder, why can't we just use the SOS operon? This is because eukaryotes don't have operons. Our genes can only be transcribed and translated one at a time. So what does that mean when our DNA breaks? When there's a break in DNA of a eukaryote, there are a lot of proteins that are needed to fix it. First are what is known as sensor proteins. After these bind, other proteins that are regulatory kinases will be then recruited. Once these bind, they send signals out. These signals are picked up by proteins that are regulatory. Once these regulatory proteins bind, the effector proteins will then bind. These proteins are responsible for the downstream effects, which include halting the cell cycle. This is done by phosphorylating certain proteins that progress the cycle. These proteins can also induce apoptosis, or cell death. Another function of these effector proteins is to induce the DNA repair systems, which is what we will talk about now. Now let's look more closely at double-stranded breaks in DNA. There are two major mechanisms to fix this. One is non-homologous end joining, and the other is homology-directed repair. Before we dive deeper into both of these mechanisms, it's important to know the systems in which these are used. There are three different kinds of systems in which the DNA repair mechanisms have been found. The two most closely related are talons and zinc finger domains. When a double-strand break occurs in either of these systems, two proteins, known as FOC1, will bind to both strands of the DNA. Once these bind, the talon system is very sequence dependent, whereas zinc finger domains will have specific binding domains that will bind to the DNA. Each of these systems will then use non-homologous end joining or homology-directed repair in order to fix the DNA. There's another significant system that is used, CRISPR-Cas9. When there is a double-stranded break in the DNA, an endonuclease known as Cas9 will then bind the DNA. It will use a guide RNA in order to find complementary sequences and target the DNA at these sequences. Discovered in bacteria, this system has revolutionized genetics and genetic engineering. Now let's take a closer look at non-homologous end joining. When there is a double-stranded break, a protein known as Q will come and bind. These will then send signals out for proteins known as DNA PCKs to bind. Once these bind, it will allow the Q proteins to phosphorylate other proteins. More DNA end processing proteins, such as the nuclease Artemis, are recruited to the DNA ends. DNA ligase 4 and its accessory proteins will then create a complex that ligates the DNA strands. Non-homologous end joining is known for small insertions and small deletions. This designates it as more error-prone than HDR, which we will talk about next. Homology-directed repair is different from non-homologous end joining in the sense that it uses a template strand to repair DNA. When a double-stranded break occurs in DNA and the homology-directed repair is used, a sister chromatid or a homologous chromosome will be used as the template strand. A sister chromatid is identical to the strand, while a homologous chromosome is almost the same. Using a template strand results in less errors and better replication. Now let's take a look at HDR up close. So here are two homologous chromosomes, and in one of them occurs a double-stranded break. First, an exonuclease will come in and degrade the 5' ends to expose the 3' ends. In order to get complementary sequences with the template homologous chromosome, this could lead to extreme sequence loss. Once these complementary sequences are found, the homologous chromosome will open into a single-stranded complex. The strand from the damaged DNA will then invade the complex and form a heteroduplex. By using the template strand, DNA synthesis will occur and repair one strand of the damaged DNA. The newly synthesized strand now serves as a template strand for the other strand to be repaired. Although this chromosome is now fixed, it can result in different sequences of the DNA. So, why do we care about these DNA repair systems anyways? Well, there are a lot of reasons, but one of the reasons is for a concept known as gene conversion. Typically, HDR of a double-stranded break uses the sister chromatid as a template strand, but if it uses a homologous chromosome to repair itself, then there might be different genes located on the homologous chromosome. So when HDR is used to fix this double-stranded break, this new gene will then be replicated where the DNA damage occurred. Although these homologous chromosomes are very similar, this new gene combination can lead to more genetic diversity within the individual. HDR machineries have evolved into a mechanism known as homologous recombination. In this mechanism, maternal and paternal chromosomes are inherited. In the event of homologous recombination, one of the maternal chromosomes and one of the paternal chromosomes will cross over. This leads to equal, reciprocal genetic information being exchanged. This concludes our discussion about DNA damage and repair. References used for this presentation were the textbook for the class, as well as a Spark page made by students in Dr. Conkle's past classes, known as CRISPR-Cas9. Thank you for watching this video, and if you have any questions or comments, please leave them below.